Cellular reprogramming offers the promise of youth, but also the threat of cancer. But what if one factor could deliver the former without the latter? Hey everyone, welcome back to Decoding Longevity, where we break down the science of aging and longevity. I'm your host, Raghav Sehgal, and I'm here with my co-host, Macsue, Jacques.
Hello, everyone. Last episode, we discussed the incredible work by Dr. Varun and all about using blood as proxies to aging biomarkers. But today's guest is Dr. Paulo de Lima Camillo, lead author of an exciting new preprint titled A Single Factor for Safer Cellular Rejuvenation. Lucas is on an ambitious journey to cure all diseases related to aging by reprogramming biology through artificial intelligence.
And Lucas is a great friend and a collaborator, and I have to say he has done some amazing work and graduated from some amazing institutions, including University of Oxford, University of Cambridge, where he is in the middle of studying medicine, as I understand. And previously, Lucas has also done his Bachelor's of Science with honors in biochemistry and molecular biology from Brown. Lucas, welcome to the podcast.
Thanks, Raghav. Thanks, Macsue. I'm very happy to be here.
Thanks for coming. So, Lucas, you have a very impressive list of publications, and this last preprint, your findings are nothing short of a very bold step. So, trimming rejuvenation down to one single gene, what sparked that idea? What's the story behind and what's rationale behind this paper?
So, I think the best way to begin the explanation goes back to a landmark paper published in 2006 by a Japanese researcher called Shinya Yamanaka. So back then, he realized that if you put 4 different genes into pretty much any cell, you can transform them into pluripotent stem cells. So stem cells that can differentiate into virtually any cell type in the body. And back in 2012, he was awarded the Nobel Prize for this discovery. But more recently, in 2019, we actually started analyzing, and just as a field in general, what actually was happening to these stem cells under the induction of these 4 pluripotency factors called OSKM. And amazingly, people notice that over the course of about 3 weeks, you can go from a cell that is aged, let's say 50 years old, up until pretty much age 0 or even slightly below 0 over the course of 3 weeks, which is this remarkable feat. It's almost like alien technology. As far as I remember, this was the first time that we were actually able to very clearly see age reversal, because before that point people were talking about slowing down aging, and even pausing sometimes was something that was almost sci-fi. And at that point, we realized we could actually go back in time from our youth state just using these 4 factors. So this is incredible technology that people started studying. So after that paper in 2019, there have been quite a few other papers studying the Yamanaka factors, these 4 pluripotency factors, and trying to either optimize them or understand exactly what's happening, what's driving this rejuvenation. The problem is, back in 2007, when Yamanaka discovered this cocktail, genes were not optimized for rejuvenation. They were optimized for pluripotency. So rejuvenation was, a very nice side effect of inducing pluripotency. So there are all sorts of issues that come with this differentiation, especially when you put these genes in mice. So if you overexpress continuously for a long time, the intestinal cells and liver cells, they quickly start losing their function, and within a few days, the mice essentially just drop dead. If you do it continuously, but not for such a long period of time, and you just stop, sometimes there is a good chance that you might develop something called a teratoma, which is this very bizarre type of tumor, which is essentially a body without a body plan. Now, if you do it just the right amount of induction, and then you stop, you see some positive side effects from doing this in mice. But the issue is that it's a very narrow therapeutic window. So at Shift, we've been trying to find different cocktails that can recapitulate the rejuvenation without inducing the side effect of pluripotency. So rejuvenation without changing cell identity, which would be sort of the holy grail in the field. When we started looking for these other cocktails, a few years ago when I joined Shift, we had this assumption that we would need to have, at the very, very least, two genes to drive a similar rejuvenation effect as with the Yamanaka factors, just because biology is very complicated. And when you start working with different combinations of genes, the space that you're exploring grows up exponentially. So we were thinking that it's probably going to be this extremely difficult task of exploring a combinatorial space of literally hundreds of millions of combinations of genes in the genome. And we started tackling this problem back in 2021 I joined. Again, initially we had this assumption that we would need a cocktail to rejuvenate cells. But over the last 4 years, we realized that there were some genes that were popping up in multiple combos. We're testing different combos and there were some overlap. The obvious idea after seeing this is let's just test the single gene that is showing up across these multiple different combos that we seem to have found that were promising. And lo and behold, we've actually tested that gene by itself. And it seems to be the primary driver behind this rejuvenation that we saw with multiple different combos. So it's something that was completely unexpected. So we came to this problem assuming that we would need probably 3 genes. With the Yamanaka factors, there are 4 genes, but some people have shown, David Sinclair and others, that 3 genes seems to be enough to drive rejuvenation. So we were never expecting that a single gene would be able to have such a noticeable effect. There have been some other papers at this point that have explored single genes. So there have been a few from the Church Lab, and there was one from UCSF as well. But none of those other papers have shown such a large magnitude of methylation age reversal. So they've shown different hallmarks of aging, so senescence, replication, autophagy, and whatnot. But when it comes to methylation clocks, so epigenetic age, which are quite difficult to change, this is the first paper with a single factor that has shown this.
Yeah, that's really exciting, and it almost seems like sort of a little bit of chance that you guys landed on this single gene instead of sort of the multiple set that you were initially planning. And so SB000, as you're calling it, turned out to be your winner. Would you actually, again, I know there's some amount of confidentiality involved here, but could you maybe talk a little bit about how SB000 works? Is it a transcription factor or a protein? And how does it really sort of quantitatively compare to the OSKM cocktail?
Yeah, so it is a transcription factor. So it is a protein coding gene. In terms of the mechanism, we, to be completely honest, we don't really know much at this point, and we're trying to figure it out. So one thing that we know for sure is that it doesn't seem to act through the same pathway as OSKM when it comes to induced pluripotency. In the paper, we also tested OSK individually, so the single genes that make up the cocktail. And it seems that whenever you add even a single one of the factors and you look at the transcriptome, you start to lose a little bit of identity. Whereas with the gene that we use, so at SB000, the identity is pretty much maintained for fibroblasts and for keratinocytes. So we also looked at some signatures of pluripotency, so beyond OSKM. Well, there are several ways that you can derive sort of pluripotency scores looking at gene ontology and different pathway databases. And for all of them, there was no increase in pluripotency with this gene. So we don't know exactly how it's working. We know it's not through the induction of pluripotency. That's for sure. We also looked at methylation and specifically at AP1 binding sites, at PRC2 binding sites. So AP1 is this complex of different transcription factors that recently has been hypothesized to drive some of the aging process.
Mhm.
And it seems that SB300, when you look at those specific methylation sites, inhibits the binding of AP1, which is something that is beneficial, supposedly beneficial. Now, there's this other complex called PRC2, which is typically very active during early embryogenesis. And Vittorio Sebastiano and Madi Mokri, they had a paper in, I believe it was Nature Communications from about 1 year ago, creating this almost universal clock. It's called the PRC2 index. And they measure the methylation at all of these PRC2 binding sites.
Mm.
And something they noticed is with different rejuvenation strategies, particularly with OSKM, you have demethylation of these PRC2 binding sites. Now, a big question in the field is whether rejuvenation is at all possible without sort of opening up these sites which are sort of euchromatic or open during early embryogenesis. And you can see this with OSKM. But interestingly, with SB300, it seems that those sites were actually hypermethylated,
Mm.
which would go in a direction which is the opposite of what you would expect. But at the same time, the interesting thing is this is more evidence that SB300 is again not acting through some pluripotency induction pathway.
Mm.
So it's not opening up these regions of the genome which are very open during early embryogenesis. So again, we still have to narrow down the mechanism, but
Yeah.
this is something that we're looking into. And when it comes to the potency of SB300 compared to the ammonic effectors, it seems to be roughly comparable to OSKM in the transcriptome.
Mm.
And when it comes to the methylome, we mainly compare SB300 with OSK, which is the sort of safer alternative to OSKM. For some clocks, it was about the same magnitude of rejuvenation. If you look at, Grim H2 and Dunedin Pace, it was about the same.
Yeah.
And some other clocks, it was actually like a much more pronounced effect. So
Mm.
if you look at, the PC Horvath, the most tissue clock, PC Skin and Blood, it was quite a meaningful change. So this was in fibroblasts. And we also tested in another cell type, in keratinocytes, which is from another germ layer, trying to assess generalizability. And to our surprise, it worked even better in keratinocytes.
Mm.
So the delta that we got was even bigger than the delta that we got in fibroblasts. And yeah, so something that is amazing as well is that when we got the data, at the back of my mind, I was sort of expecting a negative result
Mm.
for the keratinocytes. And the reason is when you looked at them under the microscopes, so we didn't show any of these images in the print, maybe for a second version, because a lot of the data that we're producing is very, very recent. But when we looked at the images from the microscope for keratinocytes under SB, and you looked at normal control cells, like I couldn't tell a difference. Like they look exactly the same. If you
I see.
just, show to people blinded, like, you couldn't tell which one was a control and which one wasn't a control. So I thought surely nothing is happening. Maybe we found a very nice fibroblast specific factor that somehow is able to rejuvenate, skin tissue, but it's not generalizable. And
Mm.
then we got the methylation results back.
Mm.
And I think for, PC Horvath, the delta was like minus 12 years or
Oh,
13
wow.
years in 6 weeks for a PC clock, which is something quite large.
Yeah, no, that's very, very interesting. And I guess it just highlights how we are our biggest critics and, we're always skeptical of all our findings as scientists. And we're the first to question, like, is this actually really true? So, yeah, it's amazing work. So you mentioned as well, like working with DNA methylation, and we know that you have an incredible paper with Raghav as well on the CpGPT. And there's obviously some validations that you're looking at DNA methylation. But I'm curious to why did you specifically started this paper or this trajectory to find this specific factor using the transcriptome? What was the sort of like rationale behind it?
Yeah, so the main rationale is that currently, if you think of a gold standard aging clock, the first thing that comes to mind to most people are epigenetic DNA methylation aging clocks. So they probably have the best correlations with chronological age, but also if you look at the mortality, there haven't been many studies comparing DNA methylation aging clocks with some other omic-based clocks head-to-head for mortality predictions. But one thing that we know for sure is that they do predict disease and mortality quite well across many different cohorts. The problem with DNA methylation clocks is that they're based in a bulk assay. So you need to collect a lot of cells. Well, it's not a ton, but whenever you're trying to do, hundreds of different perturbations, it starts to become a bottleneck to actually doing the experiment. So price per sample is another issue, and also the throughput of the assay itself. So, you can always throw money at the problem, but, when it comes to the scalability of DNA methylation clocks based on this bulk assay, it's a bit difficult to cover a very large portion of the perturbation space in different genes. So the rationale was to create some sort of transcriptome-based clock that was not only good at predicting bulk level age, but also single cell age. If you look at a lot of the transcriptomic clocks that have been created so far, a lot of them, they use this aggregation technique called metacells. So you just essentially get different single cells and aggregate them together so that you reduce a little bit the sparsity of the data. It's almost doing pseudobulk in a way, so it's not quite to the level of doing pseudobulk for the whole sample, but it's also not quite at the proper single cell level. So the main advantage of having a proper single cell clock is each cell, when you put a gene, becomes almost like an individual experiment.
Yeah.
So you can literally pair a transcriptome with a cocktail of genes, if you think about all the different combinations that you can do, and get an age measure from that. So the main rationale of creating this single cell aging clock was to increase the throughput of our experiments.
Yeah. In some ways, that's very intuitive, but also sort of really cost effective, as you pointed out, that you guys did this. I guess one sort of question going back to sort of the OSKM cocktail, and one of their biggest sort of drawbacks was the rise of teratomas, right? When we do sort of reprogramming of these cells. And you say that it seems like SP000 is not going through the same pathways, right? But could you talk maybe about a little more about the safety, the profile of this gene, and what have you tested, or have you seen any issues at this point?
Yeah, for sure. So far, we've only tested in vitro cells. So, this is a big caveat because there's quite a big jump going from in vitro to in vivo.
Right.
But in vitro, we've been able to analyze the transcriptome and the methylome for signatures of safety. So more specifically, for the fibroblasts, we looked at some genes which are specific to fibroblasts, collagen and some other ones. And it seems that all of these transcriptomic signatures of identity are pretty much maintained with SB000, which is in contrast with OSKM and OSK. And when it comes to the methylation as well, we looked at the whole methylome and tried to find samples from other tissues which resembled the methylome of SB000 treated cells. And it seems that they pretty much always clustered within the same cell type.
Right.
So fibroblasts close to fibroblasts and keratinocytes close to keratinocytes. So this is just looking at the methylome and the transcriptome. Now, something else that we did as well, which is a more functional way of measuring safety, is with something that is called a colony formation assay. So you can culture cells under this special coating, under this, special media. And if you induce OSKM and OSK for that matter as well, you start to see these pluripotent colonies, just these little blobs of undifferentiated cells just growing in culture. With OSKM, we see that all the time. It's very easy to spot. The colonies are quite large.
Yeah.
With OSK, we also very consistently see the colonies. They're typically a little bit smaller in size. And with SB000, we never saw any colony whatsoever.
That's amazing. Yeah.
So this is another
Yeah.
measure of safety. And sort of the last thing that we did, which is a functional measure as well, is measuring the secretion of collagen, which can be seen as a functional marker for fibroblasts.
Yep.
So if you just look at the control and OSK, the amount of collagen secreted is about the same. If you look at the control and iPSCs, the amount of collagen secreted goes down dramatically.
Right.
So in iPSCs, there's barely any collagen being secreted. And sort of interestingly, when you treat cells with SB000, the amount of collagen produced actually increases. Which, at the very least, it's a sign that it's not losing fibroblast identity.
Yeah, maybe becoming younger and healthier in some ways.
Yeah, for sure. So the collagen that we measure, I believe, was collagen 1. And it's a type of collagen that is known to decrease with aging. But I think overall as well, we have to be very mindful of the in vitro system for safety and how different it is from in vivo. So even the Yamanaka factors that, work remarkably well in vitro. But going from in vitro to in vivo is quite a different beast. And the next steps, we're going to test it in mice. We're going to test it in different model organisms to see what happens.
Yeah, I guess you just answered my sort of like next question. This is all very fascinating. So, but yeah, as you mentioned, in vitro models are not necessarily a very good translation to in vivo models, and in vivo models per se in animals are not necessarily a good translation to humans. Again, so it's all very, very different, but necessary steps for safety. But I guess overall, what was the next step and how far would you say until we actually can reach such therapies for humans? I guess you only tested in fibroblasts, but there is also all those different cells in the humans. What happens to the other cells? Is that something that you looking to investigate?
Yeah, for sure. So we tested in fibroblasts and keratinocytes, which are cells from different germ layers. And for fibroblasts, we tested in dermal fibroblasts and lung fibroblasts. So from different parts of the body. But when it comes to next steps, the first thing that we're going to do is to test in different cell types as well. So more cell types than the 2 that we have already tested. The second step is to do more functional measures. So we have the colony formation assay and we have measured collagen. But the different ways of assessing function of the cell, because for a lot of people who are typically outside of the aging field, when you show them the result of an epigenetic clock or a transcriptomic clock, they tend to be quite skeptical that that's actually going to translate to any functional benefit. So something that we'll definitely do more in the future is more functional assays to make sure that things related to hallmarks of aging and sort of more specific to different cell types, we can actually recover the youthful function. And the third step is to actually test it in vivo.
Yeah.
So we're going to put this gene in mice and see what happens when it comes to safety and efficacy. So we should be getting all of that data in the next few months. Again, the ultimate hope is that we're going to get this therapy, which is a single gene we're able to deliver to everywhere in the body. It's safe enough so that we can express it almost constitutively. And, the mice are just going to live longer and better. So, I can definitely see us in the future doing a lifespan study, and that would take a few years. But when it comes to the sort of next steps, testing more cell types, more functional measures, and doing safety studies in vivo are what we're planning.
Yeah. It's clearly you have a bunch of steps lined up for you, and seems like very exciting times ahead for both you and for Shift Biosciences. So with that, Lucas, I want to thank you for joining us and sharing your insights about this very cool work that you're doing. And thank you.
Yeah. Thanks for our listeners. You can read Lucas' preprint on bioarchives. We'll drop the link just below in the show notes. And thank you for joining us at the Coding Longevity Podcast. I'll see you next time for some more exciting science. Thanks, Lucas.
Right, thanks, Raghav.
Thank
Thanks,
you.
Macsue.
Thank you.